All right, so now we are at regulation of the cell cycle. So how do cells know when to divide and when not to? Sorry, I just hate when it's silent. I like the fan on in the back. Okay. So we've talked about how cells know when to differentiate or not. Now let's talk about how cells know when to divide. So the cell cycle is controlled by, I'm just going to like that, is controlled by a chemical control system that starts and stops events in the cell cycle, okay? And there are two ways or two areas that the cell cycle can be regulated from, okay? And the first is external regulation. And this can be a signal that comes from outside of the cell. So this could be a hormone, it could be nutrients, anything from your environment essentially could externally regulate our cell cycle. And then the second kind of regulation is internal regulation. And this is a signal that comes from the nucleus. So, for example, it would come from the DNA inside of the cell. So, some instruction in the DNA coded for whatever, it coded for the need to begin dividing this one cell. Okay, so two types: external regulation outside, internal regulation coming from within, from inside. And at certain points in the cell cycle, there are some checkpoints. So a checkpoint is a critical point where stop and go signals can regulate the cycle. There is a actual checkpoint between the G1 phase of mitosis, of interphase, and the synthesis, the S phase of interphase, where it's kind of like that red light, green light game, where you're like, red light, are we good? Like, is everything okay? We're at a good point. All right, cool. Green light. And then the cell is told to continue again. However, cell division, for the most part, is mostly in the off position in animal cells if there is no stimulus present. So it's not just going to happen randomly. Its default position is going to be in the off position. So I think I said 78% of the time cells are going to be in that first phase of interphase. Okay, so something really cool, and we're going back to this word that I mentioned like once before, but I didn't really get into too much. So apoptosis. So apoptosis, like I said earlier, is programmed cell death, okay? And it's kind of like essentially a cell committing suicide. Okay, and they can receive signals internally or externally. And these are signals that activate genes that produce self-destructive enzymes. Okay, so we're expressing this gene for these self-destructive enzymes. Okay, and then I want to make sure we know what the word enzyme means, so I've included the definition. So an enzyme is just a chemical substance that produces changes without being changed itself. So essentially it's just speeding something up. It's like if you were to add butter on a slide, or like butter or oil on a slide, and then you tried to slide down it, you would go way faster than if you hadn't oiled that up beforehand. So that's what an enzyme is. It's just speeding up reactions, speeding up things from happening. So enzymes are just going to speed up self-destruction, program cell death, cell suicide. Okay, and when apoptosis is triggered, so that gene is activated, then the nucleus will kind of shrink and shrivel up and will break apart. Okay, and this is very, very important because it gives us our shape as humans. So if our cells inefficiently perform apoptosis, we can end up with what you see in this image here. You can end up with web fingers and toes during development. So it's really cool. So our cells, the reason we have this programmed cell death, especially early on, is to make sure that we have space between our fingers. So early on when we're developing, our fingers are like little balls, like just little balls up here. And through the process of apoptosis, the cells between our fingers start to die and they leave space in between. Okay, so that's what formed all of our appendages, all of our fingers, our toes, just essentially anything about our shape, because we start out very kind of circular, bulgy-like, and we, through the process of apoptosis, kind of slim down and trim everything and make it look nice and pretty. Okay, So this apoptosis goes hand in hand with cancer. Okay, so cancer is, well, first of all, I want to ask all of you, what is your experience with cancer? Have you ever, do you know what it is? Do you have some semblance of an idea? Has anyone in your family ever had cancer? I've had family members that have had cancer, and I've had loved ones that have had cancer, and so it's a very critical and important, and I don't want to say popular, but common health problem these days, especially with all of the carcinogens that we're naturally exposed to every day. Carcinogens are just things that cause cancer. So it's a huge epidemic right now. So it's really important that we're talking about this. Okay, and so cancer is what happens when mitosis goes awry. So it's uncontrolled cell division. Okay, and it happens when the regulation of the cell cycle breaks down. So somewhere, somehow, something goes wrong, and that cell continues to divide uncontrollably. And cancer cells divide much more often than healthy cells do too. So it's not that it's also just doing it when it's not supposed to. It's also doing it much more frequently. So if we remember from that slide a few slides ago, when I talked about how your liver cells only divide once every, I think it was 4 months, and talked about red blood cells, how they only divide every. Liver cells was every year, and the red blood cells were every 4 months. So cancer cells will divide way more frequently than that, and so it can become a huge problem. And so this uncontrolled cell division leads to the formation of something called tumors. And so a tumor is just a clump of cells that divides uncontrollably. So once that first cancer cell starts dividing, and those daughter cells it created start uncontrollably dividing, eventually you're going to have a clump of tumor. And so all of those cells are dividing uncontrollably. And so there are two types of tumors. The first type is called malignant, and the second type is benign. So a malignant tumor is when cancer cells can break away from the tumor and be carried to other parts of the body and organs and form more tumors. So the malignant tumors are known as the ones that are more deadly and more dangerous, because if you have cancer in one area of your body, it can break away from that tumor and be carried to other parts and spread to other areas of our body. And when this happens, we say that the cancer has metastasized. I'll say that again. The cancer has metastasized. Okay, and that's when it has spread the disease from one organ to another, and this can be incredibly dangerous and fatal if not caught early. Okay, the second type of tumor is a little bit less deadly, and they're called benign tumors. And these are when they're abnormal cells. They're just abnormal, and they typically remain clustered together. However, it may be harmless and easily removed. It just depends where that benign tumor is forming. If it's forming somewhere non-essential, like on your arm or like in your fat, just somewhere where it's not going to kill you, they can be harmless. However, benign tumors start to get dangerous when you're talking about stuff in the brain, because one wrong move when you're up there in the brain can have catastrophic effects in your life and with your abilities, your ability to see, ability to speak, read, write, think. It may be harmless, but not always. Okay, so what are the causes of cancer? So cancer occurs often due to the result of exposure to carcinogens. And like I said earlier, carcinogens are just cancer-causing agents, which are chemicals that can cause cancer. Okay, and so some examples are UV rays from the sun. That's why it's super important to wear sunscreen, because those rays are going to mutate our DNA. UV rays, smoking tobacco, getting x-rays done too frequently, all of these things are carcinogens, which are stuff that can cause cancer. Okay, and carcinogens do this by mutating our DNA, by mutating our instructions, changing them without regard for whether or not it's going to completely mess us up or not. Okay, and then there are some other random mutations and some infectious agents that can also cause cancer, like HPV. HPV stands for human papilloma virus, and it can cause cervical cancer. So It's very important to make sure that you understand and that you know and understand what carcinogens are and how best to protect yourself from them. Okay, so I just said that cancer is caused by mutations in your DNA, but what does that really mean? So, a gene mutation is a permanent change in the DNA sequence that makes up a gene. Okay, so remember our DNA is made up of nucleic acids. Those nucleic acids are made up of nucleotides, and those specific sequences of nucleotides. So remember, we have adenine, thymine, cytosine, guanine, and then the RNA. The RNA nucleotide uracil. Okay, and so the sequence of nucleotides, let's say we have A, T, C, G, if a carcinogen mutates our DNA and instead makes it A, G, T, C, that's going to cause a permanent change in our instruction booklet. One different nucleotide can have catastrophic changes and differences in how our genes are expressed. Okay, so those make up our genes. Remember, that's hair color, stuff about you, everything under the sun. Okay, and it's such that the sequence differs from what is found in most people. So, in most, quote unquote, normal to typical cells in our DNA in our genome, we will have very specific sequences and that tend to be very similar across individuals. And a mutation is something that is specifically not normal and is abnormal to the norm. And mutations can affect single blocks of DNA, so like I just said, a single nucleotide, as well as large segments of a chromosome. So remember, chromosomes are tightly wound up sequences of DNA, and so they can just affect one little piece, or they can affect huge segments of a chromosome. That can include multiple genes. So that's why different kinds of diseases have multiple different symptoms, multiple different effects, because their DNA was mutated in multiple genes. So it could be a huge section of a chromosome.